Today we're doing Lab 8, which is UV reversion frequency in yeast. This is one of the most complicated procedural labs with some interesting calculations. I'll first be showing you how to make the plates that we're going to expose to UV light, about the background of the effects of UV light on cells, and then I'll talk about the kinds of calculations and graphs and questions that you'll have to answer in this lab exercise. Let's get started. In thinking about mutation, there's a lot of different categories of mutations. There's chromosomal mutations, there's point mutations, deletion, insertion, and reversion is one kind of mutation. And it's used here for a very specific reason. What reversion means is you're starting with a mutant and you're counting the number of times that by mutation it changes back into its prototrophic form. Today we're going to be using a yeast strain, and the name of this strain is His17. It has a mutation that blocks the production of histidine, and therefore, since histidine is an essential amino acid, these cells won't live unless they're supplemented with histidine in the liquid media or in the solid media. And the reason we're starting with a mutant and counting how many times it reverts is because it's so easy to count. We'll be plating this strain on media that contains histidine and media that doesn't contain histidine. Anything that grows on the media without histidine has to be there because it reverted. And more details about that later. Let's see how this is going to be done. So in order to prepare the plates, we have to dilute the concentrated oxytrophic strain in order to make appropriate dilutions for media that contains and does not contain histidine. I've already done this. Let me just walk you through how this would happen. Using sterile technique that I'm clearly not using here, I would mount a pipette tip, having set the pipetter to 1,000 microliters, 1 mil, and I would sterilely extract 1 mil of this solution and put it into a tube with 9 mils of water. I would then dispose of the tip sterilely, take the tube, briefly vortex it to make it perfectly mixed, and then take 1 mil out of that tube and put it into the 10 to the minus 2 tube here. Adding that liquid, mixing it again, and continuing the series down to where I have a 10 to the minus 5 dilution. Now, why do I need to make this dilution? UV light has two effects on cells, and we have to account for both of these. One, it simply kills cells by creating so much DNA damage that they can't repair it all and the cells die. In order to measure the killing effect, I have to put a countable number of cells, a low number of cells, on a His plus plate. This has media in it. This has histidine in the media. And every cell I put on there that's alive is going to be able to grow. So, in order to do that, I'm going to spread 100 microliters of the 10 to the minus 5 dilution on a series of His plus plates. I'll show you the calculations later as to figure out, I want to put between 100 and 1,000 cells on this plate and on the same number on all the other plates so that I get a countable number of colonies here. Okay? And so that's what happens on the His plus plates. Now we also have a series of His minus plates. On these plates, only the cells that revert will be able to grow because they are now histidine prototrophs. So since that reversion frequency can be very low, like 10 to the minus 5, we have to put a lot of cells on the plate in order to get 1 in 100,000 to grow as a colony on here. So for these plates, I'm going to use a very low dilution, like the 10 to the minus 1. This may contain upwards of 1 times 10 to the 8 cells per mil in that. So I want to put a lot of cells on the His minus plate because only a few revertants are going to grow. And I want to put a low number of cells on the His plus plate because every cell I put on there will be able to grow. Understanding which dilution goes on which plate, now I can show you how you'd plate these cells. So this has His plus. That means I need to take a very low dilution, one that I've calculated that'll put between 100 and 1,000 cells on the plate. Let me show you how you would do this. Well, first of all, you'd have a real Bunsen burner. You'd have an alcohol dish and this cell spreader. So to make this sterile, I had it in alcohol, and now I'm flaming it so that it will be sterile. Okay? Now that sterile material, now I have to put 100 microliters of the 10 to the minus 5 dilution onto the plate and then spread it. Since I'm only using 100 microliters, the 10 to the minus 5 dilution becomes an effective 10 to the minus 6 total dilution. So I draw that up, I squirt it on the plate, I take my sterile spreader and I turn and spread evenly over the surface, and that 100 microliters is quickly absorbed. I'm done with this. That plate is now spread with the right number of living histidine auxotrophs so that 100 to 1,000 cells will turn into 100 to 1,000 colonies. Now, just to show you, on the His minus plate, I'm going to do the same kind of thing. So I'll do this in the proper order this time. Get rid of the tip and mount a new one. Now this time I'm going to take what is the concentration of the 10 to the minus 1, take out 100 microliters, put it on the plate, squirt it into the center. Now, with better technique, I'll flame the spreader and cap that since we'll be done. And now put that sterile spreader and work that 100 microliters into the surface of the plate so that the colonies and cells are not all growing up together. Now, in truth, you would be making a stack of 5 plates each of the His plus and the His minus. And you'd use the same tube and the same dilution for each of those plates. And by doing that, theoretically, you've put the same number of cells on the His plus, about 100 to 1,000. And here, you would put hundreds of thousands of cells, if not millions of cells, on each of the plate, and they'd all have the same number of cells on them. Now, if you step over to the UV box, what would happen is you and your partner would have these plates labeled for time 0, 5 seconds, 10, 15, 20, 30. You'd put the 0-second plates aside, and let's imagine that these two are the 5-second plates for the killing effect and the mutagenic effect. You'd put those into the UV box. Your partner would be timing, and they'd say go. I would take the lids off, 1, 2, 3, 4, 5, put the lids back on, and take the plates out. These now have been dosed at 5 seconds each, and you'd put in your 10-second plates, 20 and 30 as well. These would then go into incubation. They're rather slow growing. We'd grow them at about 25 degrees in the incubator for at least 4 or 5 days until the colonies come up. And with that, we would have the information by counting the number of colonies on the His plus plates to determine the killing effect, and on the His minus plates to determine how many more reverted at the different temperatures. In the last point for now, the reason we have to do these separate experiments is because the killing effect is really quite profound. At 30 seconds, you will find that there's very few colonies growing. Maybe only 1% of the cells survive a 30-second dose of UV. So what we're going to do is that we're going to apply the killing effect, known as at 99%, to the number of cells we put on the His minus plate, because their cells are getting killed as well. And by doing that transformation of information, we can get an accurate reversion frequency calculated for each of the UV doses.